Hello everyone, this is Tushar Biswas and welcome to the Study Companion. Today we are going to discuss on the various aspects of photosynthesis. We'll discuss photosynthesis in a bit detailed format and we will focus on the structures specific to plant cells only that help in photosynthesis. That is the site for photosynthesis. Okay, those structures are specific to plants and only plants and not never found in animals. It's not found in animals. It's never found in animals. Okay, I will be speaking on it. I will be drawing the diagram and I will be explaining stuffs to you all. Okay, so stay tuned. If you like it, keep watching the whole video. Keep watching the whole video. Okay, and at the end of the video I can assure you that you will have the best of the knowledge. Okay, and so this is the second video on autotrophic nutrition. Another one will come, another one, and that will be the last one. Third part of the autotrophic nutrition video will be the last. Okay, yeah, it's going to be the last because I'll be discussing the stomata part over there, stomata in a bit detailed. So I'm making a short video on this topic, hardly 20 to 30, not 30 minutes also. Okay, I'll try my best to make this a short video as much as I can to make it easier for the student. So keep watching the whole video. Of course, you won't be wasting your time over here. You would learn something out of it. Okay, let us begin. Now, focusing on photosynthesis, there are three processes to photosynthesis. Three things that happens in photosynthesis. Number one, number one, absorption of the inorganic molecules. Okay, absorption of course that is something different, that comes under transportation and something different. I'm talking about photosynthesis, it's in itself. So what is it? Absorption, but of not of the molecules, it will be absorption p, absorption of the sunlight or light from the sun, absorption of the sunlight, absorption of the sunlight by the chlorophyll. Number two, first one was absorption of chlorophyll by the sunlight. Sorry, absorption of sunlight by the chlorophyll. Okay, absorption of sunlight by chlorophyll. Okay, absorption of sunlight by chlorophyll, then conversion of light energy to chemical energy by this chlorophyll. Chlorophyll, I will come to it. How is it done? I won't be focusing on the specific parts. I won't go into that much detail because it's not utilitarian to you. It's of not utility to you. Conversion of light energy to chemical energy and using of the chemical energy to split water molecules into its constituent oxygen and hydrogen. So it constituents oxygen and hydrogen. These now will be in small letters. It's okay. Oxygen and hydrogen. Okay, the second and third one is the reduction of reduction of carbon dioxide into carbohydrates. Reduction of carbon dioxide into carbohydrates. Okay, reduction of carbon dioxide into carbohydrates. So these are the three processes of photosynthesis. These are the three processes of photosynthesis. I'll be erasing the board. Now, where is the chlorophyll present? Okay, where is the chlorophyll present? In plants, in plant cell, if you see, there are chloroplasts. The various chloroplasts. Now, how do they look? For example, I'm drawing a chloroplast over here. This is a small chloroplast. This is the outer membrane, and this took a chunk of it. This is my inner membrane. Okay, this is my outer, this is my inner membrane. This is my outer membrane. This is my inner membrane, and then in the inner membrane we can always find stuffs like this, stacked on one top of another, one on top of the other, and also linked to another. All the stacks are linked to each other. Okay, the stacks are linked to each other. Stacks are linked to each other. Okay, now what are these? This whole set, of course, in bio you always do the right-handed diagram, right-hand markings. This whole thing will be your chloroplast. No, not sorry. I'm sorry. I'm sorry for this. Whole thing will be known as a grana. Always remember these when you are marking your diagram in bio, in biology. Draw the diagram, and always the marking should be in the right-handed column, and the marking should be in block capitals. Okay, block capitals for the examiner to read it properly. This is your grana. Then one single part, one single part of a grana is the thylakoid. Single part of the grana is the thylakoid, and there are various dot. There is a ground substance. Okay, that is pretty dotted. Okay, that appears dotted. That appears dotted. Now, what is that? That substance is stroma. Stroma. Grana and thylakoid. These thylakoids actually consist of chlorophyll. Thylakoid consists of chlorophyll. Hence, photosynthesis take place in thylakoid. I hope it's visible. Yeah. Photosynthesis takes place in thylakoid. Okay. Hence the same will happen in grana, because grana is the collective term for thylakoids. And the stroma, you see, stroma is nothing but filled with ribosomes. I hope everyone knows what ribosomes are. They give rise to protein. They form the proteins. Ribosomes. Then chloroplast DNA. Chloroplast has its own DNA. Like in humans, my mitochondria has its own DNA known as empty DNA. Empty. Small m, small t DNA. Empty DNA is the mitochondrial DNA. This is something which I will be discussing in the evolution chapter. And similarly, chloroplasts have their own DNA, because that's the difference between animals and plants. In animals, we have mitochondria. In plants, we have chloroplast. So chloroplast has its own. So stroma is a ground substance. Ground substance and consists of ribosomes and chloroplast DNA. And various other enzymes which I won't be speaking of right now. So this is a basic thing. This is a basic thing. You can always copy that one down. Now, where are these present? Where are these chloroplasts present? These are minute dot-like structures on the plant cells. On the plant cells. Okay. Now, as discussed earlier, the chloroplasts are minute dot-like structures. Okay, dot-like structures I said purposely because once you are viewing it under the microscope, it will appear as a small dot. But if you zoom in, you can see the whole chloroplast which I drew previously over here. That chloroplast which I drew had a cross section in it. Okay, so you won't be finding it like that. You won't find the thylakoids and grana over there. To find that stuff, you need to dissect the chloroplast. Okay, right now let us take a cross section of a leaf and see what all are present in a small cross section of a leaf. Okay, this is a leaf. We all know that. You all know this. This is a leaf. This is a leaf. We all know it, right? But this time, when we are taking the cross section, we won't take it from here. What we'll do, we'll look it from the side. Look at it from the side. Okay. How we see? How do we see it from the side? It comes like this. It comes like this, right? Am I right, man? Am I right? Right though? It comes like this. I'm right though. Yes. Thank you. I'm taking a cross section from this side, from the side of the leaf. If this is the leaf. Let my hand be the leaf for the time being. This is the leaf. I'm taking the cross section from this side. This is my hand. This is my leaf. I'm watching it from this side. So it will appear like this. It's gonna appear like this. Now I'm going to take this part. The upper, the ventral surface as well as the dorsal surface will be present in the cross section. Okay, ventral surface. Ventral means the upper part. Ventral surface as well as the dorsal surface will be present in my cross section. Both of them are gonna be present in the cross section. Okay, present in the cross section. Present in the cross section. Cross section. These two are present in the cross section. Okay, now let's draw it. What will we see over here? Okay, first we are gonna see leaf cells, cells of the plants. Okay, with their cell walls. I hope the cell walls are visible. It should be because it's pretty huge a diagram, right? Now, these are some cells which will be lining up the upper layer. Similar cells will be lining down over here. I will be drawing it a bit later on. First, we'll have cells like columnar cells, cells that look like big columns. You know, columns you have seen in various temples and like old monuments. You have seen columns like these, big columns. Okay, I've seen it in temples and various other places. There are intercellular gaps, of course. I won't be negating that term. See, intercellular gaps are present. Some it's small, for some it's huge. There are differences. Okay. The difference is now drawing their cell walls. Now drawing their cell walls. I hope the cell walls are all visible. Now there comes cell like these. First columnar, now random like epithelial cells. Okay, cells like these come in. Okay, and these cells also have cell walls. Plants, the characteristics of plant cells is that they will always have cell walls no matter what their shape and size. Okay, always remember this stuff. That's pretty important. Sometimes you are asked in examinations just because the paper was a bit tricky. Sometimes teachers have a habit of setting up a tricky paper, right? Then you're if you don't know this type of ideas, you won't be able to answer them. Na? I hope everyone knew this. That plant cell, be it any type of cell, will always have a cell wall. And the same layer of cells again reappear. Right? But this time, but the difference is the difference is that there will be a small gap. Unlike this, over here there will be a small gap or opening rather, you can say. Opening, you can say. This opening is again guarded by cells. By cells. Okay. Now time to label stuff. I'm erasing this out. I hope everyone understood, got the feel of this part. I hope everyone got the feel of this part. Okay, time for labelings. This part, the innermost part, is known as the mesophyll. Okay, mesophyll. Now, there are two parts of a mesophyll. Number one and number two. These two sets together. Okay. What is the first part? What is the first part? Where is the other pen? Just a second. Huh, got it. These are the palisade, mesophyll, and these are the spongy mesophyll. Okay, I will be drawing the I will be changing some. I will be introducing some part in this diagram. Please wait. This is not over yet. This is the epidermis. This is the upper epidermis, and these are your lower epidermis. Okay, and this is your stomata. Stomata will be discussed in the next video. This small space, small cavity over here showing in the dotted region. The small area, not dotted region, the dotted, the small gap you see everywhere. Okay, small gap over here, small gap here, here, here. These gaps are known as these gaps are known as air columns or air cavities. Air cavities, air cavities, or you can also say air columns. Cavities would be a better term. Okay, cavities, this is a better term, unlike this. Okay, this is the whole marking, whole labelings. Okay, now coming to the details. Where are the chloroplasts found? What brought us to this part? Okay, now if you remember, let me draw it with the black part now. Black is a contrasting color, would help us. Okay. This is my cell vacuole. Plant cells, second characteristic of a plant cell, have a very huge vacuole, very big vacuole. Okay, majority of the cell space is taken up by the vacuole. Majority of the cell space is taken up by the vacuole. Though not always, but majority I'm saying. There are sometimes here and there, you can always find exceptions everywhere. Okay, exceptions are everywhere. Nothing is there without exception. And these are the huge vacuoles, vacuoles, vacuole, vacuole, vacuoles. Okay, now the vacuole, around the vacuole you will find the nucleus. If you remember the cell diagram, beside there you will find chloroplast. Chloroplast. I hope the green color is being seen. Yeah, it's seen. Okay, chloroplast over here. Chloroplast over here. Okay, not only one, there will be many. The green ones are the chloroplasts. Green ones are the chloroplasts. You will find the chloroplast in the palisade mesophyll as well as the spongy mesophyll. Okay, I hope everyone is able to understand what I'm explaining. The green color is can be seen. Yeah, it can be seen. Chloroplast, everywhere. Okay, everywhere in the cell. Now, what are these doing? They are creating their own food. They're producing their own food rather through the process of photosynthesis because they contain chlorophyll. They can undergo photosynthesis, of course. They can trap carbon dioxide. They can undergo photosynthesis. Okay, guys, I purposely didn't draw any chloroplast on these upper and lower epidermis just because they do not contain chloroplast. These over here do not contain chloroplast. Okay. Now one more thing is that there is a waxy layer situated on top of this upper epidermis because once you touch a leaf you can find it's pretty waxy. Okay, the upper part is pretty slimy. That waxy layer is known as cuticle. It is waxy in nature. Cuticle is waxy in nature. Now these greens are the chloroplasts. Okay, these are the chloroplasts. The green ones are the chloroplasts. Okay, green ones are the chloroplasts. Okay, and this air cavity, why is this required and stuff will be explained later on in the next video. Okay, so I hope everything is clear. Now, what are these two will be explained in the next video. I will label them in the next video. I will explain that in the next video. Okay, okay, don't worry. Don't worry, I will be explaining these two, what do they like, what do they do, and everything will be in the next video. And that will be the last video on this autotrophic nutrition topic. Okay, so guys, everything is marked over here. You can see where the chloroplasts are present in the leaves. Okay, these are the parts that make the food. Okay, the sunlight is absorbed from this part and they make food. And this stomata, the work of the stomata is to diffuse gases in and out of the leaf. It is present in the dorsal side. I said previous on the I said beforehand only, we are taking the leaf cross section from this side. The upper as well as the lower part will be there. The ventral as well as the dorsal part will be there. So the ventral part will contain this waxy cuticle, this part upper epidermis, then this middle part will be palisade over here, spongy mesophyll over here, then the lower epidermis, and there will be small opening in the lower epidermis. This small opening is the stomata. That stomata diffuses gases in and out of the leaf cells. Leaves. What happens? Carbon dioxide comes in through here. Carbon dioxide comes in. Okay, during the day, and oxygen leaves during the day. During the night, during the night, some something else happens which will be discussed in the next video. Okay, carbon dioxide enters, is filled over here. The cells take up carbon dioxide, just pull it in, they just suck it in. Okay, they just diffuse it in. How do they take in carbon dioxide? They diffuse carbon dioxide in them. How do they do it? They just suck it in. We have all drank fruity in our childhood. What did we do? We had taken the pack, pierced the straw, and we just sucked it out. That that sucking out of the air from the straw has resulted in the rest of the liquid, the fruity, to bulge into the straw, to come inside the straw and follow the air column that is being sucked, so the liquid is following the air now. When the air column is finished, now the liquid is only left. The liquid will drop inside the mouth, will come out of the other end, will flow into the mouth. That's how the mechanism of a straw works. It's in physics. Everyone has studied that in class 7 or 8. Okay, differences in pressure. If you haven't, please do comment it down and I will make a dedicated video for that. Okay, I will explain various stuffs, physics stuff over there. If you require it, of course, if you require it, why not? Why not? Feel free to comment it down. Okay, even if there is only one person commenting, I would provide a video on that topic. I hope everything is clear by now. Okay, carbon dioxide is sucked in. Okay, like the way we sucked in fruity, the way we sucked in cold drink through the straw, carbon dioxide is sucked in. Sunlight falls over here, then the cell take in the sunlight falls directly on sunlight diffuses. Which part of the sunlight is used? The UV rays are used. The rays of the sunlight are used. The wavelengths are being absorbed now. Wavelengths, you see, as a whole, no one can touch light. But what happens over here is that anything will have a wave. Anything will have a wave nature. If you remember, anything will have a wave nature like this. If you have seen the drawing, it is the wave, that wave of sunlight has many waves. A specific wave, a specific wave is being, I won't be naming the wave, just remember this, a specific wave is being absorbed by these chloroplasts. Okay, when the sunlight is falling, the waves, all of the waves are entering like this. Okay, all of the waves are entering like this. All of the waves, if this is a wave, all of the waves are entering like this and then flowing it out. Waves do not stop, waves keep moving. Wave keep coming in and keep moving. So while they are moving, when they are passing close to a chloroplast will catch it and absorb it. A chloroplast will catch and absorb, catch and pull. A wave is passing and catch and pull. So this wave is now being taken into the chloroplast in the presence of carbon dioxide, and this wave, what does the chloroplast do? Chloroplast will split water into hydrogen and oxygen and reduce carbon into carbon reduce carbon dioxide into its uh, sugar format, into its glucose format. It will reduce carbon dioxide to glucose, it will reduce carbon dioxide to carbohydrate form. Okay, this is the whole idea. This is the whole idea of this part. I hope everything is clear right now and there will be no issues regarding this. In case, if there is any issue regarding this, any questions, anything, please feel free to comment it down. I will clear all your doubts. Okay, till then keep studying, and the next video I will feature stomata in it, and that will be the last video on this topic, autotrophic nutrition. Then we'll focus on heterotrophic. Okay, till then keep studying, keep watching the study companion. This is Tushar Biswas signing off.